Okay, great.
Yeah, I started actually.
Yep, we're good to go. Well, welcome everyone, and I've got a lot of material to cover here, so I'll try and move through it as quickly as possible so that we can get to some discussion afterwards. And so without further ado, I'll jump right into it. These are the members of the genetics group, which is AIM 3 of the Pine Map project, and I'll put this slide up again later on in the presentation when I acknowledge the specific contributions of the particular people that contributed data or slides to the presentation. So starting with an outline, I'd like to start out with some context about AIM 3 and how it fits into the overall project objectives, and then focus on the specific genetics objective that are the topic of my presentation today. And then we'll go over some basic concepts in quantitative genetics for the benefit of anybody that doesn't work in this area. And I want to spend some time discussing an issue that I think will be important for the outcomes aspect of the Pine Map project, that is moving the research results into application as quickly as possible. I'll then cover some experimental methods and materials and preliminary results, and talk about the plans for next year and some final discussion about the challenges to be overcome in the project. So this is a segment that was included in the proposal, and I just want to highlight here the same line that I highlighted in the abstract that I sent out. About half of the pine forest in the south is planted with genetically improved seedlings, and that is a total of about 25 to 30 million acres in the southeastern U.S., and virtually all of those seedlings come from these three university industry cooperative breeding programs. So the opportunity that the Pine Map project has to affect the genetics of trees planted in the South is really tremendous. We can have a substantial impact on the genetic composition of plantations across about 50% of the pine forest lands in the southeastern U.S. if we incorporate new knowledge and new methods into our breeding programs based on the Pine Map project. So these are the two major objectives of the genetics and breeding component taken from the proposal. And I want to focus on the first one. This second one, the deployment guidelines, is a complex topic in its own right, and that will be the subject of another webinar later. But note that the statement of the first objective includes both breeding and natural populations, and that the subtopics under that first objective include both identifying specific genes that control adaptation or mitigation and enabling tree breeding strategies. So these are our two key sub-objectives. So how do these relate to the project goals overall? Well, identifying genes that control adaptation or mitigation is a way of understanding the genetic mechanisms that allow tree populations to adapt to climate change and also affect the rate of climate change. Mitigation means changing the rate of greenhouse gas emission or greenhouse gas accumulation in the atmosphere. And so that affects the degree to which plantations have to adapt to changing environment because it affects the rate of change. So methods that account for genetic variation in productivity due to climate will be critical to guiding breeding programs in the future, because the objective for the breeding programs is to produce more resilient and adaptable planting stock, and we clearly have to have methods to incorporate information out of our research program into applied breeding programs in order to make that happen. And those resilient, productive pine plantations are expected to contribute to achieving USDA's long-term goals for mitigation of and adaptation to climate change. So coming back to the outline then, we'll move along for a brief overview and review of quantitative genetics. And I hope that my colleagues who work in this area will forgive me for any sweeping generalizations that I make that overlook some of the finer details, but I do want to keep this brief in the interests of time. So here's a quick overview of quantitative genetics in 5 slides. We know that phenotypic variation, or the characteristics that we see among trees in the field, is the sum of genetic variation and environmental variation, and then there's an interaction term, which is abbreviated as G by E in the equation form. And when we're modeling genetic variation at the statistical level, we model it either as additive, which means that each additional allele or gene has a consistent and detectable effect, or non-additive, which means that the effect of a particular allele or gene is not consistent and is typically dependent on interactions with other alleles or other genes. Traditionally, in tree breeding, the models focus on additive genetic variation because that's the easiest to work with. But it's important to remember, as George Box reminded us, that all models are wrong. So these additive and non-additive models don't necessarily reflect the reality of the underlying modes of gene action. What they do is accurately, more or less accurately, capture what difference that genetic variation makes in the population as a whole. And what's been found is that additive models work very well to describe genetic variation at the population level, and that's true across many species of both domesticated and non-domesticated plants and animals, as well as microbes and a whole variety of subjects. But there's increasing evidence at the level of predicting individual phenotypes from genetic variation that non-additive effects play a more important role than additive effects in many species. And I've highlighted here just a recent publication in the Proceedings of the National Academy of Sciences that demonstrates that, I think, quite convincingly for Drosophila, which is fruit flies. So as Leslie will note, I have not gotten completely away from fruit flies because I still read the literature. Now, one thing that's important, and we'll talk more about this later, is that the number of genes that affect a trait and the size of the effect that each gene has makes a considerable difference in how we can work toward our objectives in the Pine Map program. So the two general models that I want to contrast with each other are oligogenic traits versus polygenic traits. And oligogenic traits are those in which a relatively small number of genes account for the majority of the genetic variation in the trait. So a small number in this context is typically fewer than 10 genes and sometimes fewer than 5. So there may be many more genes that affect the trait to a lesser degree, but the key issue with an oligogenic trait is that the majority of the genetic variation is explained by a few genes, each of which has relatively large effects. Polygenic traits, on the other hand, are those in which all the genes have relatively equal effects and therefore all relatively small effects. And this is the biologically realistic version of the infinitesimal model, which was proposed by Fisher in 1918, which is, for mathematical purposes, he proposed a model of infinitely many genes, each with an infinitely small effect, which of course is not biologically possible, but the polygenic effect with hundreds of genes of equal and small effect is functionally equivalent to the infinitesimal model. Now, why does this matter? Well, when we're thinking about detecting the effect of a single gene, it's easier if that effect is fairly large. So if we look on the left side here, oligogenic traits where genes have unequal effects, these percentage values are just an example of the proportion of genetic variation. So the size of the font is intended to reflect that gene A here affects 50% of the genetic variation. These 4 genes each account for 10%, and these 2 genes here have very small effects and account for only 5%. Now this is a cartoon of a single chromosome with only 7 genes, and this is obviously not realistic, but the intent is simply to demonstrate the idea that there's a few genes with large effects and more genes with smaller effects. But the effect of gene A here can actually be detected in an experimental population simply by plotting the genotypic value of individuals that have 0 copies of the big A allele, or 1 copy, or 2 copies, and we see an increase in phenotypic value as the number of copies of the A allele increases, and that would be characteristic of an oligogenic effect. If we have a polygenic trait where all of the genes have equal and small effects, then it's very difficult to detect whether the big A allele is present or the big B or the big C. All we can really say is some individuals have relatively poor performance and others have relatively good performance, and we infer across that distribution that there might be differences in the number of copies of good alleles. But of course the challenge is that we can't really count the good alleles because we don't know what they are. There are hundreds of genes that might have good alleles, and so it's very difficult to detect how many good alleles any one individual has. But what we can do is look at populations of related individuals and calculate the proportion of genes that are shared in common between pairwise combinations of individuals in that population. And if the phenotype is genetically determined, then individuals that share more genes in common will be more similar in phenotype. That's a measure of how strong an effect there is of genetic variation on phenotypic variation. If related individuals are quite similar in phenotype, then the trait is considered to be strongly genetically determined. So related individuals that share many alleles in common and share desirable phenotypes are considered to have many good alleles, and individuals that share many alleles in common and poor phenotypes are considered to have relatively few good alleles. So we can rank individuals in terms of the number of good alleles they have, but we don't consider that we know anything about what genes are represented or whether or not particular individuals share a specific allele or a specific gene. And this kind of analysis has been used widely in animal breeding and is increasingly used in crop and tree breeding as well, and it falls under the general title of Best Linear Unbiased Prediction, or BLUP analysis. So if you've heard people talking about BLUP analysis in the context of breeding, this is what they're talking about. So I want to come back and focus on this question of understanding mechanism as a sub-objective versus guiding breeding programs, and talk about whether or not these are really different objectives or if they're one and the same objective. So understanding genetic mechanisms requires identifying specific genes and specific regulatory pathways that control the traits of interest. And this could include comparative analysis of data from trees with data from model plants or crop species. And the goal is to produce a comprehensive model that explains a lot of observations across a wide variety of individuals, at least, if not multiple species. And this kind of understanding has been the goal of most research that's been funded by competitive grants in the past. And it certainly has led to great advances in fundamental understanding. The objective of guiding breeding programs, on the other hand, requires developing statistical models that have predictive power within the breeding population. And it's based extensively on data from ancestors and related individuals within the breeding population, but it does not require identifying genes or regulatory mechanisms. The goal for breeders is to correctly identify individuals that will be good parents either for the next cycle of breeding or to put into a seed orchard to produce planting stock for deployment into pine plantations. So in the long term, clearly a comprehensive model of how trees respond to the environment would be a great asset to breeding programs, but that model will take a long time to develop. And we're 2 years into the Pine Map project already. We've got 3 more years to go, and we would like to have some specific outcomes that we can point to in 3 years about how we've affected breeding programs. As I talked about earlier, the easiest way to identify genes is to focus on oligogenic traits, where a few genes control most of the genetic variation, but most of the traits of greatest interest in breeding programs are often polygenic in nature. One counterexample to this is rust resistance. Resistance to fusiform rust in loblolly pine is one example of a trait that does show oligogenic inheritance in loblolly pine breeding populations, but there are few others. So I think for the 5-year term of the Pine Map project, we should consider that the two objectives of providing guidance for breeding programs versus building an understanding of fundamental mechanisms should be considered to be different objectives rather than the same objective. So moving on now to an overview of the experimental methods and materials and results. I'd like to say a few things about the analytical approaches that we'll be using towards these two different objectives. So association genetics is a means of looking for individual genes and trying to get at molecular mechanisms that control a process. And the way it works is that we take a large number of genetic variants, and it can be any kind of genetic variant. What's commonly used is single nucleotide polymorphisms, or SNPs, and we test each one of those variants for a statistical association with a phenotype using the same sort of test that I described earlier. We simply look for a correlation between the number of copies of a particular allele at that locus with the phenotype of individuals that contain that number of copies of allele. So you can think of it as a regression approach. And this works quite well for oligogenic traits that are controlled by genes with relatively large individual effects. And the magnitude of effect that can be detected is a function of the number of individuals in the population that are characterized at both the level of genetic variation and phenotypic variation. So you can do association genetics with a population of a few hundred individuals. You can do it with a few thousand individuals. In human genetics, they do association studies with populations of tens of thousands of individuals, and so they can detect very, very small effects. 1 or 2% of the genetic variation is large enough that they can reliably detect that at genome-wide significance levels. And these genetic variants, mostly SNPs, that are discovered can lead to an understanding of mechanisms. So there's no question that this is a productive and fruitful approach. But the challenge with it is that it often has a high type 2 error rate, and that is a high rate of false negative results. And I'll talk more about that in just a moment. The alternative approach is based on the BLUP analytical methods that I mentioned earlier. It's entitled kinship-based analysis here, and it estimates the relative numbers of good genes in a population of related individuals, but it does not give us any guidance on the location or identity of the quote-unquote good genes that are identified. This works equally well for both polygenic or oligogenic traits, and it can lead to predictive power. Even without the use of molecular markers, BLUP methods do have predictive power and are used in practical programs to identify individuals that will be good parents for breeding or for deployment. Now, it's important to note that that predictive power is often less than 100%. In fact, I would venture to guess it's always less than 100%, sometimes substantially less. And so there's room for improvement, especially in tree breeding programs where our predictive power sometimes is more on the order of 20 or 30% rather than the 70 or 80 or 90% that we'd like to see. So coming back to the type 2 error questions, why do we care so much about type 2 errors? Well, the reason we care is because that we're trying to identify genetic variants that are significantly associated with phenotypic variation. And a false negative result means that a gene that really is associated with variation that does play a role in controlling the phenotype is reported as not significant. And the reason for the high type 2 error rate in association studies is that anywhere from a few thousand to several million genetic variants, typically SNPs, are separately tested for association with the phenotype. And if you're familiar with statistics, you recognize that in order to achieve an experiment-wide threshold of statistical significance, you have to correct for the fact that many, many similar genetic tests have been conducted, because the standard frequentist statistical approach says that a deviation this large or larger from the expected value occurs only a certain percentage of the time by chance alone. And typically that alpha level threshold is 5%. If we're talking about significance at the 0.05 level. Well, if you test 100 genes, you expect 5 of them to be significant by chance alone. If you test 1,000, you expect 50, and 10,000, you expect 500. And so what you have to do is increase your statistical threshold of significance so that you don't become overwhelmed by false positive results. But overcoming false positive results necessarily means that you increase the type 2 error rate, that you exclude as significant genes whose effects don't surpass your elevated statistical threshold, but in fact some of those genes really do have a real impact on phenotype, and so you wind up with type 2 errors. And the classic example of this now in the literature is human height. And one of the reasons that height is particularly relevant to tree breeding is that that's a major phenotype that we measure. We care a lot about height in trees. And so the genetic basis of height growth is something that is of more than casual interest to tree breeders. And in human genetic studies, they have analyzed a set of over 30,000 individual people who have been genotyped at 2.5 million SNP loci, and did a complete association analysis and then identified about 180 SNPs that are significantly associated with height. But all 180 of those loci combined explained less than 10% of the phenotypic variation. And independent studies, multiple independent studies, have concluded that about 80% of the genetic variation in human height is genetically determined, which is to say it has a heritability of about 0.8. So a lot of genes, a lot of the genetic variation in height growth in humans is not being captured by association genetic studies. And they're doing studies that are 1 or 2 orders of magnitude more powerful than anything that we can propose to do in the context of Pine Map. So this is the basis for my concern that we will be overlooking things if we rely exclusively on association genetics approaches. So the 2 types of studies rely on different types of experimental populations, and that works out well in the context of the 2 types of populations you see on this slide. At the bottom here we have the natural population in blue, the breeding population in green. Those were elements that were highlighted in the statements out of the proposal earlier on in my presentation. Association genetics is most powerful when it's applied to groups of unrelated individuals that sample the entire range of genetic variation in the target species. And one of the resources that is important to us in Pine Map is that we have a population that was created by a previous USDA-funded project that was known by the acronym ADEPT-2. And that population consists of about 400 individual trees that are planted in a clonally replicated test planting. And there were actually 3 sites that were planted, one near Gainesville, one near Macon, Georgia, and one in Texas. And unfortunately, the year they were planted happened to be a significant drought year both in Texas and in Georgia, and the survival was extremely poor in the two sites in Georgia and Texas. So the one in Florida is the one surviving site. The trees themselves are backed up as grafts in a site in Mississippi. So we still have the germplasm, but we don't have an experimentally designed field test site other than the one near Gainesville. Fortunately, this collection of clones has already been used for collection of a variety of different phenotypic data, and more phenotypic data are to be collected as part of the Pine Map project. Now, these clonal genotypes originally were contributed by the NC State and Texas breeding programs, but the clonal genotypes that were contributed are not necessarily closely related to current breeding populations in either of those programs. So this is why it's identified in this context as representative of the natural population, is because these were first generation selections. They haven't been subjected to selective breeding or any sort of distortion of the natural distribution of allele frequencies, and they're sampled directly out of wild populations. The kinship-based analysis methods will build on current breeding populations because, as I said earlier, BLUP analysis is already used in all three of the tree breeding programs that are part of Pine Map. So those experiments will be based on progeny tests, which typically contain a larger number of genotypes, generally over 1,000 different genotypes per test site, but those are typically planted as seedling trials rather than clonal tests, which means that we have a somewhat lower degree of confidence in the phenotypic measurement of any individual tree. So that's an important distinction. There are, however, typically only a relatively small number of parents, on the order of 100, sometimes slightly fewer, sometimes slightly more. 100 parents represented by those 1,000 to 1,500 progeny. So the amount of genetic variation in the kinship-based analysis, a typical progeny test series, is actually less than in the association genetics type population, but the progeny test data are much more directly related to current breeding populations because that's where those progeny tests come from. Now, one other major consideration that we have is that we're dealing with an extremely large and complex genome. So when we're talking about genetic analysis, what we're typically looking at is variation in DNA sequence. And so it becomes important for us to consider how much DNA does the pine genome have, and what kinds of DNA is it? Well, it's about 7 times larger than the maize or the human genomes. It's diploid. Many species that have large amounts of nuclear DNA are polyploid, and they have a lot of DNA because they have a lot of copies of what's actually a fairly normal-sized genome. That seems not to be the case with pine. As far as we can tell, pine is a true diploid. The reason that the genome is so large is that it contains a very diverse collection of different types of mobile genetic elements. Retrotransposons, transposable elements, various kinds of multi-copy sequences. But unlike some cereal crops, maize is the classic example, we don't see that there are a small number of repeats that account for a large fraction of the DNA content. In pine, it seems that the repeat families are very old, they're diverged from each other, and there's no single family that accounts for more than 5% of the genomic DNA. Another issue with pine is that the genome also seems to contain a fairly high number of pseudogenes. So there was a sample study that was published a few years ago where almost 1 million base pairs of sequence of genomic DNA was determined, and the automated software programs that are used to identify genes in genomic DNA sequence identified 18 putative genes, but more careful analysis of those proposed genes by the authors led them to conclude that only 3 were likely to be actual functional genes, and the other 15 were probably pseudogenes, which is copies of sequences that look like genes, but they don't contain all of the functional elements to actually produce a messenger RNA and a functional protein. They look like genes, but they're not genes. And if we extrapolate from this sample to the entire pine genome, we multiply 1 million base pairs by about 22,000 to get up to the 2.2 times 10 to the 10th base pairs for the full genome. We expect around 66,000 functional genes and potentially 330,000 pseudogenes. So that's an important factor for us to consider when we're thinking about analyzing genetic variation, is that there may be as much as 5 times more pseudogenes in the genome than there are actual functional genes. And this becomes a problem of signal-to-noise ratio. We can analyze a lot of genetic variation, but determining which genetic variants are actually functional and meaningful is going to be part of the challenge. Now, I mentioned that DNA sequencing is the accepted method for detecting genetic variation. It's also the most cost-effective method, as I'll show in the next slide. The problem is that the pine genome is so large that resequencing the complete genome of many different individuals, as required for our population analysis, is simply out of the question. We can't afford to resequence the entire genome of hundreds to thousands of individual pine trees. So what we've been working on is comparing different methods for selectively recovering a reproducible subset of the pine genome. What we want to do is go in and get the same 1% or 0.5% or 0.1%, some very small fraction, but always the same fraction from many different individuals, and then we can sequence that subfraction, and that should give us an experimentally feasible approach for analyzing the populations that we're interested in. Now, we recognize that there may not be a single technology that's the ideal solution for every experimental purpose. We have different types of experimental populations and different analytical approaches, and so there may be differences in the genotyping strategies that are optimal for these different populations and experimental methods. This slide captures some information that's released by the National Institutes of Health. The Human Genome Resource Institute supports major genome sequencing centers in the United States, and so they have a very robust data set on the cost of DNA sequence analysis, and the short message is that the cost has dropped by a factor of about 10,000 over the past 10 years. And at the current rate, we expect that the yield of DNA sequence per dollar will double about every 7 months. And clearly this exponential trend can't continue indefinitely into the future, but it does seem likely, from what I've heard about emerging technologies, that the trend will continue for at least 2 or 3 more years. So what we can expect is that there will be, over the remaining 3 years of the Pine Map project, we may see another 10 to 20-fold increase in the amount of DNA sequence per dollar. And there isn't any other genotyping technology that could be applied in our research that has this same kind of price trend. Other genotyping technologies are available, but they tend to be getting more expensive over time rather than less expensive. So the 2 major categories of methods that we've been testing are both methods for reproducibly capturing a subset of the genome and DNA sequence analysis of that subset to detect genetic variation, but they focus on different subsets of the genome. In one case, the hybrid capture method focuses on the subset of the genome which is similar to expressed genes. And we're fortunate that loblolly pine was the subject of several experiments to detect expressed genes before PineMap was started. So there's a extensive database of expressed gene sequences, and those can be used to synthesize custom DNA bait molecules, which can be used to capture target fragments of genomic DNA that are similar in sequence to those expressed genes. So this strategy focuses our analysis of genetic variation on sequences that are similar in sequence to expressed genes. And this is where the abundance of pseudogenes in the pine genome could be something of a concern, because by definition, pseudogenes are similar in sequence to expressed genes. So we need to be cognizant of the fact that not everything that is recovered on the basis of sequence similarity is necessarily coming from a single functional gene. There may actually be a family of genes that are similar in sequence and have to be disentangled from each other. The alternative approach is to use restriction enzymes, which are simply proteins that cleave DNA at a specific nucleotide sequence, and to select a subset of the genome based on the presence of restriction enzyme cleavage sites. And by using methylation-sensitive enzymes, we can selectively enrich for the portion of the genome that has a lower content of methylated DNA bases, specifically cytosine. And methylcytosine is one of the mechanisms that is used in plant genomes to inactivate mobile genetic elements. So by focusing on the portion of the genome that has fewer methyl units, we hope, focusing on the portion of the genome that has a lower content of repetitive DNAs and therefore is perhaps enriched for functional genes and transcribed sequences. But we're not focusing on the basis of sequence similarity, we're focusing on the basis of the presence or absence of DNA methylation. So this is an overview of the SureSelect technology from Agilent, and these slides were provided by Meng Lu at Texas A&M, who has been doing these experiments. And you'll notice that the top of the slide shows two different tracks of effort. That is, the blue boxes here on the left show the process of designing the bait molecules and creating what's called the capture library, which is the collection of sequences that are to be used as baits. In the case of the preliminary experiments at Texas A&M, they used a collection of about 35,500 sequences of expressed genes that were provided by a collaborator at Miami University in Ohio, and they synthesized bait sequences at 2x coverage, so each nucleotide in that collection of genes was covered on an average of twice. This yielded a total capture library size of about 78 million base pairs of DNA. The other aspect of sample preparation is shown on the right, which is to take genomic DNA samples from the tested individuals, 1, 2, as many as you can afford, and you carry out a sequence of steps that produces fragments that are ready for DNA sequencing. And then the final stage of the process is to actually do the hybridization capture step, and that's shown in more detail on the next slide here. These are the the prepared genomic DNA samples. The red additions at each end represent the sequences that are required for sequence analysis using the Illumina technology. These are the bait molecules here. The little blue spheres represent biotin molecules, which can be used to selectively recover those DNAs. So the two populations of DNA are allowed to anneal to each other, which means that they form double-stranded, base-paired, double-helix DNA molecules, and then the capture probes are selectively captured on magnetic particles that contain streptavidin, which binds very strongly to the blue biotin spheres. And so the capture probes and any DNA molecules, any genomic DNA molecules that are annealed to them are selectively captured by that process of hybridization and then capture onto the streptavidin magnetic particles. You then denature the DNA again, recover the genomic DNA fragments identified by the red Illumina sequencing adapters on the ends, and you carry out your DNA sequencing process. So that was done with a pair of samples, and the samples were both taken from a single seed. So it was a megagametophyte, which is a haploid tissue, and then the diploid embryo from the same seed. And the haploid DNA content of the megagametophyte represents the maternal contribution to the embryo. So the megagametophyte sample represents one allele of every gene, and the embryo has two alleles of every gene. They may or may not be different, but you can see that the sequencing experiment yielded extensive coverage, 80 to almost 100-fold coverage of the 78 million base pairs in the capture library. This means on average each of those 78 million base pairs should have been detected in 85 to 96 different sequence reads on average. So the megagametophyte reads were assembled into contigs, that is short for contiguous sequences, so almost 185,000 different contigs. So you'll notice that the 35,500 different putative expressed genes were not recovered by that assembly. Ideally, the de novo assembly would have returned the same number and the same sequence as what was used for the original bait design. But the total size of the contigs that was recovered is slightly larger than the total size of the target sequences that were used for design. So there seems to be ample coverage, even though it's not completely uniformly distributed. Fairly conservative parameters were used for SNP detection. There's more detail here than we have time to talk about. But the important observation down here is that about 30% of the embryo reads aligned back to the megagametophyte assembly, which means that about 70% of the reads did not. So the question of how much of the reads obtained by this type of approach are on target, that is, map back to the target portion of the genome that we're trying to sequence, and how much is off target, is an important factor for future optimization experiments. The coverage of the 35,000 unigenes was pretty consistent, 92%. Only about 2,600 were not covered at, this is at high mapping stringency of 99% similarity. If that's dropped down to 95%, then there are only 755 unigenes that didn't have any reads assigned to them. And if those 755 are aligned against the draft loblolly pine reference genome sequence, 168 of them don't align to the reference genome. So it's not clear where the problem is. If it's a problem with the analysis that produced the express gene sequences, that these 168 genes really don't exist, or if the problem is with the draft loblac pine reference genome, and it's really too early to say. Both of these projects are still under development, and there will be more data within a year or so, so it will be easier to resolve these questions in the future. Coming back here to the alternative approach, the restriction enzyme-based methods, run through that fairly quickly. We tested two alternative approaches. The initial one that was developed back in 2008 used a single restriction enzyme, and that cuts genomic DNA at these sites marked with the black X's, and so-called barcode sequences are attached to different samples. So individual 1 gets attached to one barcode, individual 2 gets attached to a different barcode, and then all of the different samples in the experiment can be mixed together, which means that we're not handling hundreds to thousands of different DNAs at that point. We may have only a dozen different samples. And the DNAs are randomly sheared then to break the genomic DNA fragments and create another end so that the fragments are of the appropriate size for Illumina sequencing. And this process is a little bit more technically complicated and a little more expensive than the alternative, which is to simply use two different methylation-sensitive restriction enzymes. And you digest, you can use the same restriction enzyme with the black X's, but you use one that cuts much more frequently, and therefore you reliably produce a large population of molecules that are of the appropriate size. Now, there will be some molecules that are too small, as shown by the box marked A, and other ones that are too big, and so you don't recover those when you do the selection of fragments of appropriate size. But the fragments that are selected in principle will be more uniformly and reproducibly represented in all of the different individuals that are being analyzed in the analysis. So just to come back to our two general methods, we have the hybrid capture and the restriction enzyme results, and there are two subsets of the restriction enzyme, random shearing versus the double digest with two enzymes. So a quick overview of the hybrid capture. The Agilent SureSelect platform was used for this, but there are other platforms that are available. So we have a total of about 78 million base pairs of bait sequences based on the sequence of 35,000 genes. Recovered genomic sequences similar to most of those target genes. The overall level of putative SNP variation is consistent with what's been identified in coding sequences of expressed genes in previous studies. So after filtering, there were a total of about 51,000 SNPs that were reported. Most of the sequences out of the embryo were non-target sequences. About 30% of the embryo sequence reads mapped to the target region, but most of the target genes that were identified did have relatively complete coverage. So based on these results, we can say this method is a relatively efficient way to resequence the coding regions of many genes. Some genes don't seem to work well with it. It remains to be seen if optimization of the bait designs can overcome that problem, or if there will always be a small fraction of the population of genes that simply fall through the cracks in this approach. The random shearing approach is called RADseq. This was carried out by Jason Holliday and his co-workers at Virginia Tech. They analyzed 2 diploid DNA samples and 10 megagametophytes that were taken from seeds of one of the 2 diploid parents and produced a set of sequences, but many of the sequences contained artifacts of the adapters that were used in library preparation, and so they are not really useful. So the decision was made in a conference call that we would focus our attention on the double digest and hybrid capture methods, that there was not worth continuing with the random shearing approach. So the double digest method, we carried out a similar kind of analysis, only we expanded the number of haploid samples because we had some additional seed on hand. So we had 2 diploid DNA samples and then 90 megagametophytes from seeds of one of the diploid parents. We identified about 47,000, a little bit more than 47,000 sequences that were present in about half of the megagametophytes, which is what we expect for a single allele at a single gene. That allele should be present in half of the individuals, and the alternate allele should be present in the other half. So we had about 47,000 candidate markers that met that criterion. About 70% of those markers aligned to the Pine reference genome sequence, and about half of the ones that mapped would align only to a single location in the draft reference genome sequence. So those are our candidate single copy genetic markers. 95% of the candidate single copy markers aligned without any gaps, which means that there were only nucleotide polymorphisms in the alignment. And the majority of those were 1 to 4 candidate SNPs. And this is out of a 64 base tag. So anywhere from 1 in 64 to 1 in 16 nucleotides is polymorphic. This is a much higher rate than has been described in coding sequences, but that's one of the advantages of sampling from genomic DNA without limiting the search to coding regions, is that the rate of polymorphism is expected to be higher. This is a linkage map that contains 529 of those candidate single copy markers. The number of recombination events in this population is limiting, so there's not room for a whole lot more markers. There are a few large gaps here where we could expect to see more markers inserted in the linkage groups. There is also another smaller fragment of a linkage group that probably belongs on the end of one of these smaller groups of the 12 major groups here. But overall, this is fairly good evidence that markers that appear to be single copy actually are segregating and behaving as Mendelian single loci in this sample of genomic DNAs. In terms of functional genes, about 17% of the 47,000 candidate marker sequences align to sequences in the draft Pine transcriptome assembly, and that identifies about 5,000 different sequences in the transcriptome assembly. Again, most of those alignments are ungapped, so there are SNP variants between the sequence reads and the transcriptome reference sequences. And simply by chance, about 62 different map locations on the preliminary linkage map correspond to markers that align to transcriptome sequences. So it does seem that we are identifying some functional genes and that we can map those functional genes on genetic maps, but it's not exclusively focused on functional genes. It's simply sampling from functional genes. One important issue: chloroplast DNA is not methylated, and so it will be completely digested by the methylation-sensitive enzymes. And so there's always a concern that a large fraction of the sequence reads may be derived from chloroplast DNA. That turns out not to be the case. Analysis of the entire population of sequence reads showed that it was less than 1% that were derived from the chloroplast genome, and only 375 of the 47,000 candidate markers were derived from chloroplast DNA. So that's not too much of an issue. So plans for year 3. We need to reduce the per sample cost of the hybrid capture approach, and Meng has experiments underway to try and do that. I think there will be some experiments underway at University of Florida that are aimed at trying to reduce that per sample cost. And so that will be a valuable contribution. The 400 trees that were sampled from the range-wide distribution will be genotyped using the hybrid capture protocol, and the same trees will be genotyped using the double digest GBS procedure, so that we have 2 different genetic marker methods on the same population of trees. And this will allow us to test in more direct comparisons the merits of these two approaches, one focused almost exclusively on regions of the genome present in expressed genes, and the other portion of the genome that is sampled more or less at random from the non-methylated or relatively low levels of methylation subfraction. So that would give us two different kinds of markers and two different subsets of the genome for use in the association analysis. We'll also be testing kinship-based methods on progeny sampled from field tests in the breeding program, and genotyping samples on the order of 1,000 to 1,500 trees per site for 2 or more sites with different climate characteristics, and including those genotypes in kinship-based analyses. And we will be interested to see how many SNPs we get in expressed genes with this approach, but that's not really a priority because the objective is to use the genetic markers to make inferences about relationships among individuals. So we're not necessarily trying to make associations with specific expressed genes. And the lower cost and higher throughput of the GBS procedure are likely to be major advantages. So we've got some important questions to answer. We don't know how many markers we need to make a difference in the accuracy of our kinship-based BLEP methods. We don't know how much of an increase in accuracy we can obtain by including the marker genotypes. There are some alternative strategies that we'd like to explore. If we're genotyping thousands of progeny that are descended from only dozens to perhaps 100 parents, one option is to genotype the parents very, very intensively and then genotype the progeny at a much lower density and simply infer or impute the missing genotypes from the parents where we have full information down to the progeny where we have a sort of a scaffold or a skeleton map. And that could lower our costs pretty substantially. And then another question, of course, is if we discover genes in the ADEPT2 population by association genetics, how easily can we integrate that information into the BLEP analysis in the breeding population? So we've got some challenges to overcome. We're going to be dealing with a literal tsunami of genotyping data, terabyte quantities of data. I mentioned the challenge of signal-to-noise resolution a little bit earlier, and we have to make decisions about which types of data will be appropriate for solving specific kinds of problems. And so that's an issue that we'll be working with as we make our way through these genotyping objectives. We also need to think about collecting appropriate phenotypic data in breeding programs. So the tests that we're using for the purposes of PineMap are tests that were planted but are fairly unusual with respect to the distribution of genotypes outside the range where they're considered to be well adapted. And we need to think about whether the breeding program should be incorporating this type of testing more extensively into their field plantings in the future so that they can be collecting data relative or relevant to the question of adaptation in the field in the future. And then, of course, we have to communicate the results clearly to stakeholders that will be making their own decisions about how much risk they choose to take versus the potential reward from planting material that may or may not be well adapted in the region where they're planting. I want to acknowledge again the contributions of the people that actually did this. I mentioned Jason Holliday, and his group carried out the RADseq. Meng Lu worked with Carol Loopstra and Kostya Kutowski at Texas A&M to carry out the SureSelect. And the work at NC State was done by Laura Townsend and Will Colway, working under my direction. And Gary Peter is working at University of Florida on some hybrid select capture experiments as well. So with that, I'll close. Just 1 minute over, but I'd be happy to take any questions or any further discussion if there's an interest. So thanks very much.
Hey Russ, there's a question coming up from the UF Pine Map crew that they're typing in. So looks like there's.
Yep. Okay. Yeah, I'm watching. Well, one of the things that's going to happen, I've been working with a draft genome assembly that was released in about June, I think, and that assembly was based on what's called a whole genome shotgun. So it doesn't include as much information as is available about how different parts of the genome relate to each other. And I've talked with David Neal and with Steve Salzberg, who's working on the assembly part of the project, and they tell me that they will be releasing another draft of the assembly that will include much more information on genome structure, and that will probably be released sometime between now and the Plant and Animal Genome meeting in January. And so I anticipate that one way in which PineMap can work more closely with the reference genome project is to incorporate that newer assembly as quickly and as seamlessly as possible into our analytical pipelines, and also to provide feedback to the reference genome project on any abnormalities or ambiguities that we detect in our genetic analyses. So we will be working with large populations of very diverse individuals. We will have more information than is available to the Reference Genome Project in terms of potential population issues and variation within or across the population in DNA sequence. And so we can perhaps provide some guidance to them in identifying areas that need particular attention and could be strengthened in their presentation or in their assembly process. So I'll be giving a presentation at the Reference Genome Sequencing Workshop. There is a workshop on Saturday before Plant and Animal Genome, and so I'll be meeting with them, and we'll be discussing ways in which we can work more closely together to try and integrate the information that we have that might be useful to them, as well as integrating the information that they have, which would be useful to us.